Hello and welcome to another Bio43 pre-lab video. This video will provide some background on enzymes and inhibitors, which you'll later apply in designing another experiment. You may have already heard some of this in lecture, but I want to make sure everyone begins on the same page. And even some biology textbooks include misunderstandings about inhibitors that I want to clear up. Let's start with free energy, usable energy that can do work. All molecules have an inherent free energy, and a reaction is thermodynamically favorable if the free energy of the products is lower than that of the reactants. A reaction will only occur spontaneously if the change in free energy, or delta G, is negative. However, just because a reaction is spontaneous doesn't mean it will occur in a reasonable time frame. Some reactions essential for life would take tens of millions of years on their own. That's because they have a high activation energy. In order for the reactants to be converted to products, they must first pass through a very high energy transition state. And the higher the activation energy, the slower a reaction will occur. Enzymes are proteins that catalyze reactions. They increase the rate of a reaction without being altered themselves. The starting reactants and final products are the same, so enzymes have no effect on the delta G. They can't change whether or not a reaction is spontaneous. But they stabilize the transition state, lowering its energy and increasing the rate of the reaction. This is the generalized equation for an enzyme-catalyzed reaction. The reactants are called substrates, S, and they bind the enzyme, E, at a place called the active site. This forms an enzyme-substrate complex. The binding causes the enzyme to change shape such that it can form favorable interactions with the transition state. With the activation energy lowered, the substrate is rapidly converted to product P, which is released from the active site, leaving the enzyme in its starting state, ready to catalyze the reaction again. So to understand how well an enzyme functions, there are two main things we want to know about. How well does the enzyme bind its substrate, and how quickly can it convert the substrate to product once bound? The study of these things is enzyme kinetics. And a typical approach is to measure the initial reaction rate, V0, under different substrate concentrations. This gives us a Michaelis-Menten plot. As substrate concentration increases, so does V0. Linearly at first, but then it starts to level out. It approaches, but never quite reaches Vmax, the theoretical maximum rate of the enzyme. This is a rate at which the reaction would occur if after the product was formed, a new molecule of substrate bound the enzyme instantaneously. So Vmax is a parameter associated with the second part of this equation. The higher the Vmax, the better the enzyme is at carrying out this step. The other parameter we get from this sort of study is a Michaelis-Menten constant, or Km. This is the substrate concentration necessary to get a V0 equal to half of Vmax. This parameter is tied to the first part of this equation. The better the enzyme is able to bind the substrate, the less substrate will be required to reach this rate. So lower Kms indicate better enzyme function. Now there is a major problem with this analysis. To find the Km and Vmax, we need to know the equation for this curve. This type of curve is called a rectangular hyperbola, and it can be described by the Michaelis-Menten equation shown here. However, hyperbolic fits are difficult to perform without complicated software. We get around this by plotting 1 over V0 and 1 over the substrate concentration instead. By taking the reciprocals and doing a little algebra, the Michaelis-Menten equation transforms into this, the Lineweaver-Burke equation. This equation describes a line where 1 over Vmax is the y-intercept and Km over Vmax is the slope. And performing a linear fit is simple. In the next video, we'll go over the steps of collecting enzyme kinetics data, plotting it, and finding the Km and Vmax. But before we do that, I want to discuss inhibitors, molecules that decrease enzyme activity. We can learn much about an enzyme's function by what inhibits it and how. There are many ways to classify inhibitors. Irreversible inhibitors form covalent bonds with enzymes. Such bonds are difficult to break, so once formed, the enzyme is permanently inactivated. Reversible inhibitors form non-covalent interactions with enzymes. Such inhibitors bind enzymes and inhibit function while bound, but they can also dissociate, leaving the enzyme active again. Reversible inhibitors are further classified by where they bind the enzyme. Competitive inhibitors bind the active site, blocking access to the substrate. This only affects the first step of our equation. It makes substrate binding more difficult, so more substrate is needed to compete with the inhibitor and reach the rate of half Vmax, so the Km increases. But when a substrate is already bound, a competitive inhibitor has no effect on catalysis, so Vmax is unchanged. Since they are both able to bind the same site, competitive inhibitors tend to be structurally similar to the substrate. Other inhibitors bind the enzyme at sites other than the active site. These are called allosteric inhibitors, and the sites they bind are called allosteric sites. Allosteric inhibitors are further classified based on whether they prefer to interact with the free enzyme or the enzyme-substrate complex. An uncompetitive inhibitor only binds to the enzyme-substrate complex. By binding and removing the ES complex, uncompetitive inhibitors actually drive the first step of the equation forward, leading to a lower Km. This is a bit counterintuitive, since we said a lower Km is associated with better enzyme function. But the enzyme-substrate inhibitor complex won't convert the substrate to product. So it doesn't matter how well this first step is functioning when the second step is blocked. Due to their effect on the second step, uncompetitive inhibitors lower Vmax, which is more like what we'd expect from an inhibitor. Some inhibitors can bind the allosteric site for both the free enzyme and the enzyme-substrate complex. These are called mixed inhibitors. Even though they can bind to both the free enzyme and the enzyme-substrate complex, they often prefer one or the other, and their effect on Km varies based on this preference. Similar to an uncompetitive inhibitor, a mixed inhibitor that binds better to the enzyme-substrate complex will decrease the Km. But a mixed inhibitor that prefers the free enzyme will increase the Km. A non-competitive inhibitor is a special class of mixed inhibitor that binds the free enzyme and enzyme-substrate complex equally well. Because of this, it has no net effect on substrate binding, and the Km is unchanged. All mixed inhibitors, including non-competitive, prevent the conversion of substrate to product, and therefore reduce Vmax. All of these subclassifications have been for reversible inhibitors. Irreversible inhibitors all affect Km and Vmax in the same way, no matter where they bind the enzyme. Having an irreversible inhibitor bind an enzyme is equivalent to removing the enzyme from the reaction entirely. Any enzyme unaffected by the inhibitor will bind the substrate like normal, so Km is unchanged. But since there is less functional enzyme present than normal, Vmax decreases. This is true even if the irreversible inhibitor binds the active site. The substrate can't outcompete something that forms a covalent bond at the site. There are other molecules we could talk about, like activators, which bind allosteric sites and increase the Vmax, but we're not going to go into detail about them today. So this will do it for our overview of enzymes and inhibitors. Time to put this into practice. See you next time.